Hello everyone, today I am pleased to introduce a research on multi-omics insights into surface charge effects to decode the interplay of nanoplastics and bacterial antibiotic resistance. Nanoplastics, emerging environmental pollutants with widespread distribution, pose significant threats to ecosystems and human health by facilitating the evolution of bacterial antibiotic resistance. While impacts of nanoplastics with varying surface properties on the modulation of bacterial antibiotic resistance remains unclear. To address this gap, we investigated the roles of nanoplastics and surface charges on the antibiotic resistance of E. coli K-SHAR, focusing on the underlying molecular mechanisms. This study provided valuable theoretical insights into how nanoplastics with distinct surface properties influence the evolution of bacterial antibiotic resistance, paving the way for innovative strategies to mitigate bacterial antibiotic resistance contamination. Highlight 1. Multi-omics approaches revealed how nanoplastics with different surface charges influence antibiotic resistance in Escherichia coli. 2. Positively charged nanoplastics enhanced antibiotic resistance by upregulating genes and proteins linked to oxidative stress tolerance and efflux pumps, and promoted antibiotic resistance genes transfer via conjugation and formation. 3. Negatively charged nanoplastics disrupted biofilm formation and metabolism, potentially reducing antibiotic resistance. 4. These findings highlight the critical role of nanoplastic surface properties in shaping microbial resistance dynamics and highlight emerging risks posed by nanoplastics to public health through accelerated antibiotic resistance propagation. Based on drug sensitivity assays and changes in the relative abundance of antibiotic resistance genes, positively charged nanoplastics significantly elevated antibiotic resistance genes levels through mechanisms like target protection and efflux. In contrast, negatively charged nanoplastics likely decreased antibiotic resistance by promoting antibiotic inactivation. Using laser scanning confocal microscopy and scanning electron microscopy found that nanoplastics aggregation and membrane damage in positively charged nanoplastics treatments enhanced antibiotic permeability and efflux. In contrast, aggregated negatively charged nanoplastics formed a physical barrier between bacteria, potentially disrupting intercellular communication and reducing horizontal gene transfer or antibiotic resistance genes transformation. Transcriptomics revealed that oxidoreductase activity, energy metabolism, and responses to oxygen compounds were the main functions associated with differential expression genes under positively charged nanoplastics treatment. These functions could enhance resistance to oxidative stress and support bacterial survival under antibiotic pressure. Additionally, differential expression genes impacted cell membrane functions, such as biofilm formation, leading to weakened bacterial adhesion and reduced biofilm-associated resistance, particularly in the presence of negatively charged nanoplastics exposure. These findings were further confirmed by proteomics analysis. Bacterial growth curves indicate that positively charged nanoplastics-induced apoptosis may cause DNA replication errors or impair repair mechanisms, thereby promoting adaptive mutations and resistance. Genome resequencing detected an indel variation in the positively charged nanoplastics treatment, which was associated with hydroxylamine reductase HCP, an enzyme involved in nitrate-nitrite reduction and oxidative stress response, may reduce intracellular oxidative stress and aid antibiotic resistance. The whole genome sequencing showed that positively charged nanoplastics promoted plasmid replication or enhanced the acquisition of free plasmid, potentially promoting vertical transmission antibiotic resistance genes. Conversely, plasmid length decreased in negatively charged nanoplastics, indicating plasmid loss, possibly due to reduced cell cycling and metabolic activity. Meanwhile, we developed conjugation and transformation models and found that positively charged nanoplastics exposure significantly increased conjugator abundance and transfer frequency. And this phenomenon is also clearly manifested from E. coli to environmental bacteria. These suggested that positively charged nanoplastics promote antibiotic resistance genes dissociation via enhanced horizontal gene transfer in E. coli, raising concerns about increased resistance spread when such particles interact with environmental microbes. High concentrations of nanoplastics significantly increased antibiotic resistance in E. coli K-2. Positively charged nanoplastics significantly increased antibiotic resistance by enhancing oxidative stress tolerance and antibiotic efflux pump activity. Negatively charged nanoplastics inhibited resistance by disrupting bioformation and metabolism, potentially forcing bacteria to lose resistance as a survival strategy. Positively charged nanoplastics also promoted both vertical transmission and horizontal gene transfer of antibiotic resistance genes, escalating the risk to human health.